record. And it'll record it, it's going right now, and then it's done.
And then there's this one, which is about the same, I guess. All right, I guess we'll start. I guess many of you took up my promise of trying to videotape this lecture. I guess there's not many. If the camera were to zoom around, you'd see there's not too many of you here. But those are, I mean, I guess those who are here agree with me that Slopely doesn't really begin until the afternoon. And I hope this lecture is worth your attendance. I think it should be. So we end the semester with endemic and epidemic diseases and a few examples. So the epidemics are often defined as being either what is called host-to-host or common source epidemics. These are common source epidemics, and we've seen a few of these already. Not so much epidemics about them, but at least we've heard of cholera, and EHEC E. coli doesn't really have a name yet, but it's something you've seen before. You've seen the toxin involved in botulism, and so on. So we'll come back to this at the very end of the hour. But I think first we should look at a few examples. This is not working because this is not on. There. Of epidemics that are far more lethal, way more lethal, and they're invariably spread from host to host. They can be counted. These are just some examples. Some things you've heard of some of these before. Ones that are transmitted by coughing and sneezing from person to person. We've heard a little bit about influenza. We've heard a little bit about whooping cough, tuberculosis. We haven't really touched on very much. Diphtheria, really just the toxin. Also, sexually transmitted diseases. Of the many diseases, these are the four that you hear most about. Well, these are the ones we're going to talk about. And there is also ones that are born from person to person via an insect or something like an insect. So plague has been a huge killer. It is transmitted by fleas. So of these, we'll focus initially on, I guess pretty much exclusively, on these four. Diseases that are sexually transmitted. So if you've ever gone to the Planned Parenthood people and asked for an STI test, what do they test you for? Does anybody know? The standard test is for gonorrhea. It's probably if you have the handouts. I uploaded these very late. If you wanted to print them out, I apologize, but they're up on the blackboard. The standard test is for gonorrhea and syphilis and chlamydia and HIV. So for all we hear about other STIs, STDs, what is the difference between an STI and an STD? I think it's kind of an important distinction. Anybody? Oh. Well, I mean, an STD, the D is disease, and that means that a person feels sick. An STI means the person has been colonized but may not feel any symptoms. And that's an important distinction. And we'll see that there's a huge gap between an STI and an STD. Many people who are infected feel no symptoms. And that's important. Okay, so for all you hear about other STIs, for example, herpes and human papillomavirus causing genital warts, the Planned Parenthood do not regard those as serious public health threats. That generally the consequences are fairly limited. These are the four that they seem to care about. It also may be that human papillomavirus is just a lost battle. If you're sexually active, you probably have it. And as I say, except for cervical cancer, the consequences are pretty mild compared to some other diseases. Of course, there is a new vaccine, which is it's terrific, but we don't have time to talk about it. So let's go on and talk a little bit. We have one slide about each of these three bacterial diseases, and then a bunch of slides about HIV and AIDS. Okay, so first of all, let's back up here. These are the three bacterial ones. Neisseria gonorrhea. If you've ever, these are the proteobacteria. In here somewhere is E. coli, that's a gamma proteobacterium. Agrobacterium is an alpha. This is the beta group. So it's a beta proteobacterium. It's not too far from E. coli, but not too close either. Another beta proteobacterium would be whooping cough, pertussis, Bordetella pertussis, which maybe actually be up here, I don't know. Nope. Chlamydia is, it's in its own group. It's a deep branch. These are all the eubacteria, all the bacteria, not the archaea, but the bacteria. It's in its own deep branch. Treponema pallidum causes syphilis. It is called a spirochete. It's a member of a spirochete order, which a spiral thread is what spirochete means. So there's a number of other ones that are you sometimes hear about. So let's talk a little bit about each of those. One slide each. Okay. Treponema pallidum. Pallid, pale yellow. Treponema, twisted thread. It is a spirochete. That group also includes Borrelia burgdorferi, which causes Lyme disease. They have this spiral morphology, and it's odd. They have a periplasm. They have an inner and outer membrane, and between the two membranes, this is a cross section. These are flagella. And there they are in a cartoon version. These flagella are endoflagella. They are flagella that live inside the two membranes in the periplasm. And I have to admit, I have a hard time getting my head around this, but these evidently, they each one of them can spin the way flagella do. They're like a propeller. They spin each individually, and somehow that makes the whole thing, first of all, corkscrew looking, and also the movement is kind of a corkscrew movement. And I just can't quite understand how these individual flagella can do that, but they do. So this spiral shape is all due to these endoflagella. They are not culturable in vitro. Now that's a theme we'll see in the other two bacterial STDs, STIs, the bacteria we're going to talk about. They're not culturable. So that to me is a huge, it means a lot, I think. It means that they have been undergoing an obligate symbiosis in that broad sense, any kind of interaction, for a long time. They are obligately symbiotic, which is a funny choice of words, but it's correct, as endosymbiont. They've probably been co-evolving with their hosts, either human hosts or animal hosts, for a very long time. And we see this in a lot of pathogens that are obligately tied to their host and they cannot replicate outside the host. They often have undergone a lot of regressive evolution. They've lost genes. Their genomes get smaller. So that is a small genome. Maybe you don't have a sense yet for this, but the biggest bacterial genomes are about 12 megabases. The smallest ones are less than a half a megabase. An average one is about 5 megabases. So this is definitely on the small side for a bacterial genome. And if you look at if you were to analyze the genes that this thing encodes, you'd see it's missing a lot of genes that you would think would be very important by regressive evolution. There's evidence that there was evidence for and evidence against, but I guess the best evidence is for an origin of this bacterium in the New World. So perhaps, that Columbus brought lots of plagues to the New World and he brought this back with him. If not Columbus, then someone after him. Okay, so there is you hear a lot about these 3 stages of syphilis. Primary syphilis causes now I used to call that a canker, but that's not right. A canker is a more general term for a sore. The word chancre, which is the correct pronunciation, is reserved for a big, ugly sore that is characteristic of primary syphilis. And I was not going to show you a picture of that because they're too disgusting. But they do resolve spontaneously. And then the person is symptom-free for years. Years later, you get a secondary, I hope your patient gets a secondary, or maybe your patient doesn't get that because they've been treated by then. But if untreated, years later there can be a systemic rash, which is also not life-threatening. Tertiary syphilis happens later than that, and that can be life-threatening. But it's interesting, this is... one of the points I want to make is that these have been evolving for a long time. They are serious medical problems, and yet they don't kill you immediately. They kill you very slowly. And they have learned to live with us. And in many cases, not so much this one, but the other 2, remain asymptomatic. Let's come back to that in the next slide. So that's all I want to say about Treponema pallidum, because we have a lot to cover actually. Next one, chlamydia, the most common STI in the US. Now this is an important point. In the textbooks and the websites, they talk about, well, what are the consequences? What are the consequences and how do you treat it? But in fact, they just in passing mention that 75% of women don't have any symptoms. And that to me is important. I mean, if someone has symptoms and you want to treat them, then maybe you want to focus more on the symptoms. But if you're taking a more detached view about the evolution of this interaction, it is important to realize that many infected people have no symptoms. And I would go out on a limb and say that you might consider this as having a little flavor of a commensal pathogen. We talked a lot about Staph aureus and Mycobacterium tuberculosis and Streptococcus and Helicobacter that can cause disease, can cause serious disease, but can also, in other people, be completely asymptomatic. And I would tend to think that this is kind of similar. It sometimes causes no consequences, and we should keep that in mind. Those who do have symptoms have symptoms. This is important. Once again, here we go. Another example of a very small genome. Regressive evolution, loss of genes, and that suggests strongly a long-term, millions of years of coevolution with a human host or a non-human host. It is like Treponema, it is also unable to replicate outside of a human cell, of an animal cell. There is this is from the textbook, it talks about how there is this form called an elementary, I don't know why it's called elementary, but it's called an elementary body, which is metabolically inert. It's almost like a virus. And it can invade a cell and replicate. It can differentiate into what is referred to as a reticulate body. Once again, I don't know the source of that word. The reticulate body is the replicating form, and they only happen inside of a host cell. And then they do replicate in a vacuole, and they can escape. They can differentiate back into an elementary body, which then can escape and infect other cells. So all replication occurs inside of human cells, and you can't, no matter how much you try, you cannot culture these things anywhere else. So once again, the strict endosymbiosis suggests an ancient association and a lot of coevolution. Last one, Neisseria, which causes gonorrhea. As I said before, it's a beta proteobacterium. And again, only 20% of women have any symptoms. I hope that's right. If you wait enough years, I could be wrong, but that's my reading of it. At least for extended periods of time, many people have no symptoms. So once again, this has, I hate to say it, kind of some of the character of a, a commensal pathogen, something that can be pathogenic and also can be commensal and cause no harm at all. It has a pretty small genome. It can be cultured in a petri dish, but with difficulty. It's much happier inside of a human cell. It's almost as though if you waited 5 million more years, it would evolve into something that is obligately intracellular, can only grow inside of an animal. But it hasn't got that far yet. It has 2 megabases. It can grow on petri dishes, but it is referred to as fastidious. This word is often used to describe a strain of bacterium or a fungus or anything that is difficult to grow. If you're going to grow this on a petri dish, you require, what they do is they take agar and they add a whole bunch of nutrients, but they also add red blood cells. And then they autoclave it and they look kind of brown, so they call that chocolate agar. But it is not chocolate, it is red blood cells. But the point being that you need, whatever is in red blood cells seems to be important. They need a lot of amino acids and vitamins and God knows what else. Okay. An important thing about this bug is one of the reasons it's so successful is that its cell surface keeps changing. What's the name of the lizards that keep, chameleon. It's like a chameleon. It has a number, this is the best described example. It has a pilus. It has a type 4 pilus. We've seen type 4 pili a lot. What's unusual about this type 4 pilus is the following. It has a single gene that has a promoter and has the full length pilus gene and makes the pilin protein. So it has one gene like that. It has actually 12 what are called pil-S, S for silent. These are partial copies. They don't have a promoter. They're never expressed, but they have a good bit of the DNA required for a functional gene. They, by a mechanism that is still kind of mysterious, it can recombine from this part of the chromosome into the expressed copy of the pilin, of the pilus gene. So there it is. The silent copy is going to undergo a homologous recombination. They have a very strong sequence similarity, and they undergo a recombination event where the information that was here is lost, and the information that was here is now being expressed. And it can take more of this gene or it can take less of this gene. Now, what can then happen is maybe PIL-S2 can do the same thing. A few weeks later, in a subset of these bacteria, perhaps just one bacterium, this can be recombined into this locus, and you get a new flavor of pillin. And it does that often enough that just as the host is beginning to make antibodies against the pillis, you get a new pillis. And so it's, like I say, like a chameleon. It's changing its colors. It's changing its antigenic determinants. Other proteins that are expressed on the outer surface of the bacterium do the same. Well, it's not the same. The details are not the same, but the net effect is the same. They keep making a new flavor of all these proteins. And so they keep evading the immune system. Also, they express a fairly famous protease called IgA protease. If you think about how this is going to be colonizing the urinary tract, the genital tract, if you think back about IgA, IgA is the only secreted antibody. So it's the one that Neisseria has to worry about, and it has a protease that can destroy IgA. So it's another way that it can evade the immune system. And I think that's all I wanted to say about these three bacteria. HIV. So we'll have a fair amount. It's a fascinating history. HIV. An HIV infection causes AIDS, obviously you know that. This is an interesting graph that's telling us a lot. That upon exposure to the HIV virus, the virus replicates rapidly. There's no time, there's no y-axis here, but it replicates rapidly to a high titer initially, and then it falls down to almost, now this is, where they show a y-axis, it's usually a log axis, so this really could be just about zero detectable virus. And that's because you get a strong antibody response. You get a lot, now GP120 is one of the outer membrane, it's an adhesin, it's in your notes somewhere. It doesn't really matter. It's one of the proteins of HIV. It happens to be one on the outer surface that helps it to adhere to host cells. You get a lot of antibodies against it. So the body, your patient, the infected person is doing a great job of making antibodies against it. It's also making these T helper cells that we heard about before. It's making a lot of those that are specific to HIV. So the immune system is working beautifully. And it's almost wiping out the virus. The virus falls way down to almost undetectable levels. And then over a period of years, sometimes 12 years, we get this gradual loss of the T helper cells. We remember now that the virus targets T helper cells, which are also called CD4 cells. The receptor, the adhesin, actually that adhesin binds to that receptor, the CD4 receptor. So these cells are the target, and they eventually become depleted. And the virus eventually goes up, but this does take a decade. At some point, then, we do get a severe loss of T helper cells and a severe increase of virus, detectable virus. And then this lucky person underwent the cocktail, and his or her viruses went way down to nothing, and the CD4 cells went back up again. If you don't have antiretroviral therapy, then you would otherwise get, oh, you've heard about, opportunistic infections, lots of different fungi and bacteria that wouldn't normally harm anybody suddenly become threatening. One good example, especially in the developing world, what happened, I keep hitting this button, is tuberculosis becomes a huge problem in HIV patients. This word, seroconversion, is an important word. So what this word means is that if a person is infected for a number of days or weeks after infection, focusing on the blue line here, there are no detectable antibodies. And then on a certain day, suddenly the antibodies begin to be detectable. So when a person does not have detectable antibodies, that person is called seronegative. And if a person has antibodies, they are called seropositive. And if the interval in between, they seroconvert from negative to positive. And that is diagnostic of having a disease. In many diseases, you know, if you're going to be tested for HIV, they do not test you for the virus. First of all, it's expensive, and second of all, it's not as sensitive. What they test you for instead is antibodies against the virus. And so if you are seropositive, if you have antibodies against the virus, that is considered to be diagnostic for infection. So you do, but it also tells us that the patient that has HIV does make gobs of antibodies against it. It's a very successful, almost very successful response to infection. And it almost wipes out the virus. But you remember, HIV is it's the one, of course, that copies its RNA into DNA, and the DNA integrates into the genome. So even though the virus may make no virus RNA, it's still there. It's still in the human DNA, and it's being carried passively, analogous to a lysogenic bacteriophage. It's being replicated passively as the host genome is replicated. And eventually it becomes reactivated. Okay. Early history of the AIDS epidemic. I went on the web and took some of the most important dates off this. A person in the Congo died in 1959, and decades later, and some tissue, blood, I suppose, was saved. And I suppose they must have dried it, because I can't imagine it was refrigerated for that long in the Congo. But anyway, something was preserved from this patient. Decades later, diagnosed as having HIV. That was done by PCR, which of course was invented in the 80s. But they would go back to this old tissue, this old sample, and diagnose that it had HIV in it. And the person apparently had symptoms that resembled AIDS. 1959, way before it became a headline. An estimated arrival of HIV from Africa to Haiti. So from the US, it seems that it came from Africa to Haiti to the US. And then here, these are all done by extrapolations. Probably it came to the US in about that date. So in 1969, somebody died in St. Louis. Somebody died of something that resembled an AIDS kind of diagnosis, immune deficiency. And many years later, that was diagnosed. Some sample of blood was diagnosed as having HIV. So the first infections began to happen about that date. And then in 1981, this is 13 years later, we begin to see an epidemic. 121 deaths by the end of the year. So that's when I became aware of it. Here, it doesn't matter. They gave it a new name. Before that, it was originally thought to be a disease of gay men. But it really never was a disease of gay men. It was a disease, in the early days, there was the term, the 4 H's. The earliest victims were either homosexual or they were hemophiliacs, because of course hemophiliacs get injections of blood clotting factors, and they were contaminated with HIV. Or they came from Haiti, because the disease came from Africa to Haiti to the US. Or they were drug injecting, they injected drugs recreationally. Or 2 of the above. So the 4 H's. So this early name, gay-related immune deficiency, was replaced by the current name. 1983, people in France isolated the virus. Hemophiliac, first death of a hemophiliac because of transfusions. And 1986, Anthony Fauci? I don't know. I read the name, but I don't hear it. Estimated 1 million deaths, 1 million infected people in the US. So then we have a real full-blown pandemic. Okay, so that's beginning at 1959, but we actually go back farther in time. If you go back farther in time, the feeling is, the evidence is overwhelming that HIV, well, first, it's closely related to another virus, it's called SIV. It's called SIV for simian immunodeficiency virus. But in fact, it is, I should stand in front of the camera so we can, right, there. Yes, so we call it the simian immunodeficiency virus, but of course it doesn't really cause very serious, I think it's the next slide actually, does not cause serious consequences. SIV evolved into HIV probably more than once. The estimates are 6 times. Where did it happen? Well, back to that 1959 victim. That 1959 victim died in a city, Kinshasa, which was Leopoldville, in what was then the Belgian Congo. Okay. And you can go back to, you can sequence HIV, and you can go and find, this is the most serious world pandemic. And you can go back to ask, of all the different SIVs, which ones most closely related to, which one was most closely resemble the pandemic strain? And where are they located? Where geographically are they located? And those, that's been done, and those are located in what is now the Congo, which was then the Belgian Congo. So the best estimate is that the species jump, the evolution of SIV into HIV with a concomitant jump from a chimpanzee or a gorilla or a monkey, probably a chimpanzee, into a human, occurred in what was then the Belgian Congo, and it happened probably around 1908, plus or minus a decade. That person here, it's the cut hunter hypothesis. The idea is, of course, people make jokes about how someone must have had sex with a chimpanzee. That's unlikely, because for one thing, they are nasty. But for another thing, that's just, there is this bush meat trade where people in that part of the world eat chimpanzees and monkeys. I don't think they eat gorillas, but anyway, they eat great apes. And the theory is that somebody up in the hinterlands, way up in wherever in the Belgian Congo, was either cut or bitten by a chimpanzee that was being harvested for consumption, and at that moment, just about that instant, SIV evolved into HIV, because SIV does not infect people, but HIV does. So there was a species jump with a little bit of rapid evolution right then. And that would have been up in the hinterland somewhere, and then perhaps that same person or someone that he had sex with or something like that traveled down a river to what was then Leopoldville, is now Kinshasa. But that word Leopoldville is important. That name, so at that time, the Belgian Congo was almost owned privately by the King of Belgium, King Leopold. I think that's, I forgot about this. The SIV, the bush meat trade. These are slaughtered carcasses. I can't recognize them, but I suppose they're monkeys or maybe chimpanzees. The important thing here is SIV and these apes have co-evolved for how many years. SIV is not a serious problem in apes and monkeys. For many years, it was. They thought it had no consequences. If you look in the laboratory, they don't look sick at all. But then people began looking in the wild, and apparently in the wild, yes, there's more opportunities for infection, and they do suffer some immunodeficiency, but not nearly as severe as humans. Now, this is a very important point here. So remember we talked about how we often diagnose people as if you've been infected if you have antibodies against a certain pathogen, that means you've been infected by it. And it's easy to take a blood sample and look. So people in this trade, people that actually butcher and sell this kind of meat, if you test their blood, many of them are seropositive for SIV. So they don't have any disease at all, but they have been infected by SIV, and they mounted a fully effective, complete, successful immune response. And they wiped it out, and they're perfectly healthy. But they have been exposed to it, and they have antibodies to prove it. So it seems as though the transmission of SIV from apes and monkeys into people has probably been going on for hundreds of years, but they were fought off. The immune response did its job, and these people were healthy. So in epidemiology, there's the disease triangle, the host, the pathogen, and the environment. The environment is incredibly important. So back in the back in the day, 1,000 years ago in Africa, people were living, I suppose, in tiny tribes or what have you, and if there was an evolution of SIV into HIV, it might have killed one person, but it didn't spread. Because human habits were not conducive to the spread. And that changed. That changed in about 1908. So we said that King Leopold was important. In about this time, There was what was referred to as the scramble for Africa, that if you look at the continent of Africa in 1914, you can see that virtually every bit of it was colonized, was a colony of a European country. Somehow Ethiopia managed to kind of delay that until Mussolini in the 1930s. And the other one is Liberia, which you may know was basically a US colony, sort of. It was nominally independent, but it was set up by, after the Civil War, for the idea of repatriating African Americans. Never really caught on, but that's almost American. And that managed to be independent for a couple more decades. The rest of it is all a colony of Europe. And in here is the Belgian Congo, and here in 1914 was Leopoldville. And this was a time of incredible turmoil throughout the continent. This, right? So there was, the European colonists were rapidly industrializing, a lot of roads, a lot of railroads, a lot of agriculture, a lot of minerals, rubber sap. People were moving around, they were getting displaced from their aboriginal homes, they were being urbanized, cities were growing rapidly, populations were going up. In a more urban environment, there could be prostitution. You had, largely male labor camps where they might engage in prostitution, or they would perhaps use the services of a prostitute. A lot of disease and malnutrition. And this was the background for that species, not so much the species jump from apes to humans, but that emergence of a pandemic. The human behaviors that foster the spread from person to person of this disease. Also, there were a lot of people were getting sick, and vaccines were becoming very popular, and antibiotics also. A little later, antibiotics. So there was unsafe use of needles, not recreationally, but by doctors. Back to Leopold. There is King Leopold. He wasn't a very nice guy. I don't have much information about that picture, but it kind of says a lot, right? And it's a photograph. It's, the 20th century, not the 19th century. Well, it could be the 19th century. But this was an interesting practice of King Leopold. He demanded a certain quota of rubber sap from people in the Belgian Congo, and if they didn't deliver the quota, he would have their hands chopped off. So that's kind of symptomatic of the kind of behavior that the Europeans were doing to the natives in Africa at that time. And it may be facile to conclude that this caused HIV, but that turmoil and the new behaviors, this urbanization, the slavery basically that happened in the early part of the 20th century had a huge amount to do with from what could have been a single case of HIV, of AIDS, becoming a pandemic. It had a lot to do with these new kinds of behaviors that people were engaging in, largely against their wills. So that's kind of a dark idea. All right, let's go on. We would like to think, I keep hitting the wrong button. We would like to think that there'll be a vaccine or something else will happen to make HIV go away. And I don't know why it's taken so long. But in the absence, say this happened 10,000 years ago, before anyone had any notion about medicine, would we, we said also that SIV has evolved for a long time in apes and monkeys, and it's pretty benign, fairly benign. Could we imagine a day when HIV would be less severe than it is now? Could that ever happen? The answer is yes, it could happen. This you have seen before, or you saw a picture like it. This is the virus, and it has an adhesin, and the adhesin binds to these two receptors, CD4. This is a helper T cell, has a CD4 protein on its surface, and it also has the CCR5, a co-receptor. In the cartoon, you can see the little complementary shapes here. So CCR5 is a co-receptor for the HIV virion. Turns out that there are people out there that don't have this protein. So in the cartoon down here, CCR5, this is a person who has two good copies of CCR5, and makes lots of the CCR5 protein, and is fully susceptible to HIV. But a surprisingly high number of people, Caucasians have a particular deletion of part of that gene, and they don't make a functional protein. Actually, it's a deletion of 32 nucleotides in the gene. Now, 32 is it's going to delete 10 amino acids, and it's going to throw it out of reading frame, so you make no functional CCR5. They have fewer CCR5 proteins on their cell surface, and they are still susceptible, but they progress from infection to symptoms to having AIDS more slowly than people that have 2 good copies. And there are people that have no good copies, and they are completely resistant to this. So you have these 2, if you're homozygous for the lack of this, it's fantastic, because you don't have to practice safe sex. So anyway, these people, of course, there's other examples of human genetics that have been evolved in response to pressures from pathogens. And of course, the classic example is the sickle cell anemia gene in response to malaria in African populations. And of course, if you have one bad copy, then you are protected against malaria and pretty healthy. If you have 2 bad copies, you have sickle cell anemia. Another good example of that we haven't talked about is cystic fibrosis, which 2 bad copies of that gene causes cystic fibrosis, which is a terrible disease. 1 bad copy and 1 good copy causes no symptoms of cystic fibrosis and protects against things like cholera. So we didn't talk about that, but that's another good example. This is also very interesting because the people that are deleted for both of these genes, if you had 2 bad copies, they're perfectly healthy. They may be a little more susceptible to something, but they're perfectly healthy in day-to-day life. And they are completely resistant to HIV. This, you don't have in your notes, but this is a map of the incidence of that mutation. And this 0.13 means 13%. 13% of people in certain parts of Europe and Central Asia they are heterozygous. They have 1 bad copy and 1 good copy. So about 1% of these people have no good copies. So that's fairly high. Unfortunately, that gene is unknown in sub-Saharan Africa. But you could get the idea that, that if this pandemic were to go on for another, for thousands of years, and it was to hit the whole world as badly as it's hitting Africa, that there would be a strong selection for this mutation, or some other mutation, something like it, that would make people completely resistant. So yes, humans will or would evolve resistance against HIV. There are, I'm not going to go through this, it's way too busy, but you get the idea that there are many other, here's CCR5, if you are homozygous, you are resistant to infection. If you are heterozygous, you progress to disease more slowly. There's a lot of other genes that, certain alleles of which make people more or less susceptible to HIV. And the resistant alleles would be selected for. And if you wait enough centuries, enough millennia, humans would evolve to be more resistant to HIV than they are today. We hope that we'll have a vaccine way before that. But if we don't ever get a vaccine, we can look to evolutionary pressures to alter gene frequencies to a more resistant, the humans may evolve resistance. I think that's all I want. Is there anything else about it? No. Okay, so now just a few words about infectious holocausts in the New World, in the North America and South America. We're all familiar with how Native Americans fell in droves to infectious diseases at the hands of the conquistadors in Mexico and South America. So this is a picture, I don't know who drew this, but that person presumably has smallpox. A Native American presumably has smallpox. A book was written about this that used to be, one year was the required reading for incoming freshmen, called Guns, Germs, and Steel. And basically it should have been called Germs, Germs, and Germs. Because the idea was that the Native American immune system was no match for European germs. We Europeans were germier than they were, and our immune systems were stronger. And it had a lot to do with Native Americans did not domesticate very many animals, very few. I forget there was like 5 in the entire 2 continents. So they really didn't domesticate very much, and whereas the Europeans domesticated a lot more animals, and we had a lot of more exposure to nosocomial infections, and we had a stronger immune system. But that's Central America, or Mexico and Central and South America. It was just as big. I think it's not as obvious to many people that was just as important in New England. 100% as important. So this is a great map. This map was drawn by somebody under the leadership of Samuel du Champlain, a French explorer in 1606. This is the harbor that became Plymouth, Massachusetts. And it was a thriving, I don't know if these are, what are those, trees or houses? These are people. The idea is that this was a thriving Indian village. What became Plymouth, Massachusetts was a thriving Indian village in 1606. And there was a big plague in 1616 to 1619 in this part of North America. And it wasn't just Plymouth, it was the entire Northeast. By about this time, well, between these two dates, there was a lot of trading going on. I guess if they'd already, the French got to Quebec a lot earlier, I think. Forget when exactly, but, well, maybe about this date. And there was a fair amount of trade, a lot of fishing going on. Europeans were kind of visiting the coastline by about that date. But the Indians were still very abundant in number. And then there was a huge plague that wiped out a huge number of them. This is the title of a paper that would appear in Emerging Infectious Diseases. A quote from that: estimated deaths range from 1/3 to 90%. I have read 96%. Plymouth was severely depopulated. So just like, who was it? The conquistadors in Mexico. Just like that. It's exactly the same story. We don't know what the plague was. This paper proposes that the disease might have been leptospirosis. It doesn't matter. These are other theories as to what that plague was. We don't really know. These are symptoms, and I guess they think the symptoms are most consistent with leptospirosis. Like I said, it doesn't really matter what the disease was. But New England was severely depopulated by epidemic plagues, just like Mexico was. And not just New England, but all the way across this country, this continent. The European colonizers were, they would come in peace and just shake the hand of the Indian tribe on the next hill over, and a month later, 90% of those Indians were dead. And that was the reason, really, that North America is not populated by Native Americans to this day. In large numbers. If you look, I'm sure Europeans would have loved to wipe out all the natives in Africa. It didn't happen because Africans had better immune systems. So really, germs were the reason that this country is not more populated by Native Americans than it is. Okay, sometimes these were done. Oh, this is John Winthrop. He was the first governor of Boston. Before he left England, he arrived in 1630. The year or two before he left, he wrote kind of a treatise about, is it right or is it wrong for Europeans to colonize the New World? And he writes these sort of questions, and he answers them. And the answers are supposed to be persuasive. And one question is, but what warrant have we to take that land, which is and hath been of long time possessed by others, the sons of Adam? And the answers are, firstly, that which is common to all is proper to none, which means they don't put up fences, they don't file deeds in the county courthouse. Who says it's theirs? That was one argument, right? There's more than enough for them and us, another argument. This is the best one. God hath consumed the natives with a miraculous plague, whereby the greater part of the country is left void of inhabitants. So in other words, they're all dead anyway, right? And there's another one. So that was John Winthrop. So he knew fully well by 1629 that there was this huge depopulation by a miraculous plague. Sometimes these epidemics were done deliberately. So this is Geoffrey Amherst, a British general in the New World. You will do well to try to inoculate the Indians by means of blankets, as well as to try every other method that can serve to extirpate this execrable race. I should be very glad of your idea to use hunting them down with dogs, and so forth. So even Hitler didn't write it down. Hitler never wrote it down, but Amherst did. Hitler never wrote any of that, but he did. He didn't seem to care. All right, that's Amherst. Now that's all I want to say about host-to-host epidemics. They have a huge part in history, obviously. Let's go back to less important but sort of fun Not fun. Not fun for the victims. Common source epidemics. That was the first slide. I want to say a few words about cholera and then finish the semester with typhoid fever. Okay. So cholera in London. You've heard about maybe you've heard about John Snow. There was this certain pump, a water pump in London. And this is 1854, long before, decades before the germ theory disease was ever established. But there was this pump, water pump, and these little bars here indicate deaths from cholera in a certain time frame in London. And he made this argument that people that drank from this pump died. The ones that, the people that were close to the pump died more than anybody else. And furthermore, people who lived close but didn't use that water survived. And he made this kind of correlation that people that drank from that pump died and people who didn't drink from the pump didn't die. This is the first bit of epidemiology in the history of humanity. There's the pump. The problem is I've been to that intersection and there's no pump there. So I don't know where this pump, I've been there. And there's a plaque. And that's the plaque. The red granite curbstone right there marks the site of the Broad Street pump. So that's where the pump was. And so I don't know where this pump is. It's not there. Anyway, you see this on the web a lot. There's the pump, and it's not there. It's not true, but okay. Maybe it's nearby. The point is, John Snow thought a pump was, and they actually excavated around there. They took off the pump handle, and the epidemic went away. And then later they excavated, and they found out, yes, there was a cesspool right next to the pump, and it was not good. But this is what you hear about. This is in your textbook. But another study that is less well appreciated, maybe more definitive, was the following. There was at that time in London a number of private companies that would supply water in pipes, not just hand pumps, but they would supply water in pipes to citizens of London. And there was a number of companies that did that. And one of them was called the Southwark Vauxhall Company, and then there was the Lambeth Company. And they provided, so the Southwark Vauxhall provided water here and here, and whereas Lambeth provided water here. This region of London was provided by both. Both companies ran pipes under the ground and provided water to the people of London. They competed for the privilege of providing water in this part of London. So that is a beautifully controlled study. And Snow gathered a lot of data. And epidemiologists always complain about they need better data. We don't have good enough data. But he got what he could from this region. And he showed basically the following, that this light blue is the region of London. Light blue was supplied by this company. The red was supplied by this company. And this part of London was supplied by both companies. And the cholera epidemic was severe wherever Southwark Vauxhall supplied water and not severe anywhere else. And there was tables and tables and tables of numbers. This was actually, the numbers were reanalyzed using Bayesian analysis in the last 10 years, and that's where this picture comes from. So that was the dawn of epidemiology, kind of figuring out, correlating victims with something. What do the victims all share that they don't share with non-victims? And this was a beautiful study, and it's in all the medical school textbooks, and so there. Now we're getting a little late, but if I just have 3 or 4 more minutes, we got one more thing to talk about, and it's just fun. So, and then the room is available, so if you can stay for a few more minutes, it'd be great. This brings us back to home. These are the top 10 outbreaks of contaminated food or water, dwarfed by the number of deaths from flu and smallpox, but still among waterborne or foodborne illnesses, these are the top 10 in history in the US. And Ithaca, New York got edged out by Long Island. In 1924, there was 150 deaths. In Ithaca 1903, there were 82 deaths. 29 of them were Cornell students, and then it goes down, the list goes down from there. So let's finish the semester talking about that epidemic. This is George Soper. George Soper he had a PhD, and he's a sanitary expert for New York City. And he wrote this article. He came and analyzed the problem that had happened. He came the next summer and analyzed everything, and he wrote a paper that you can get from the library. And actually the 4-credit option people read this paper. Saying an epidemic of typhoid. And he said, epidemic of typhoid fever, which broke out, deserves to be regarded as one of the greatest outbreaks of preventable disease that ever occurred in New York State or the entire country. George Soper was an interesting guy. He also tracked down Typhoid Mary. And I was going to try to do this, if I can do it. Yeah, this is a map. Typhoid Mary was, was a cook in New York City and Westchester County, and everywhere she went, she left a trail of typhoid fever in her wake. And actually, 3 people died, so the deaths were not that terrible, but that was bad enough to get her in a lot of trouble. And they put her, not in jail, but they quarantined her away from other people for about 27 years of her life. She died in this quarantine. And the quarantine, you may want, so there's Manhattan, right? And there's an island. And if we switch over to this view, there's an island just off of Manhattan. It's actually the East River. North Brother Island. And there it is. Now, how do I make this full screen? I guess I don't. There it is. That's North Brother Island. And there's a massive hospital. Can I get even closer? That's a massive hospital that is just completely abandoned. The whole island is completely abandoned, but you could get there by kayak if you wanted to. There it is. And that was a huge hospital. Back in the days when they thought that it was really important to quarantine the infectious disease victims away from the rest of us, they used this North Brother Island. There's another building down here. So anyway, they put her there. And that was George Soper. So let's go back to George Soper. And go back. So there it is. And he came to Ithaca to investigate what had happened. And this book, a book was written just 2 years ago about this whole thing, the epidemic. It's all about this plague, this epidemic in Ithaca. And this was reported at the time, 1903. The number of cases of typhoid fever which have developed in Ithaca in the last 10 days indicate the serious epidemic, and so on. The cause of the fever is undoubtedly the city water supply. And that was what happened. Over here it says, basketball practice will be resumed at 6 p.m. will be continued daily. Yale has been playing a consistent game throughout the season and will come to Ithaca in good form. Okay, so we go a few more days later.
They're not leaving good form.
I'm sorry, well, we'll see. Here's the fever. No worries about patient privacy at this time. These are all students, very sick, no change, convalescing, very sick, slight improvement, quite sick, slight improvement, and so forth. Typhoid fever claimed a 4th victim of the students of the university in the person of George Westman, who died at the infirmary shortly before noon, and so forth. So that was not good. And furthermore, we got beat by Yale in basketball, 13-5. That's a low score. I don't know. Maybe both teams were sick. Anyway. Because actually, typhoid visited Yale sometime later, too. I mean, it wasn't just Ithaca. Another one here. The fever situation is unchanged, and so forth. These people are still sick. I guess this is continuing the first column. The following resolutions were adopted by the freshman class at a meeting in Barnes Hall, whereas it has pleased Almighty God in His infinite wisdom to take, and I cut it off there, but it's an obituary. Maybe that's not the right word, but anyway. So it was not good. And so George Soper comes and he confirmed what everybody knew, that people that drank from so there was a reservoir being constructed here in 1903. People that drank water that was flowing down this river, there was an older reservoir from which water was taken, they got sick. Ithaca students, at least on the campus, drank from a different reservoir, which is still there. If you walk up to the plantations, it's still there, and they did not get sick. And at that time, there was water being taken from Buttermilk Falls, and people who drank that water did not get sick. But people who drank from this one did. It's just like John Snow, isn't it? People who drank this water got sick, people who didn't drink this water did not get sick. It's exactly the same idea. And not that many years later. This is a picture of the reservoir that was being built. And the reservoir was not finished at that time. It was being built. And the workmen, I guess there's a workman right there, were pooping on the shore near this thing. And that is thought to be the cause of the epidemic. This is that same reservoir I should go back. This reservoir was the one that was being built, or at least the dam was being built at that time. Much later, this became a newer reservoir. That's a source of Ithaca water, and this is primarily used for skinny dipping. This reservoir in that creek there is, anyway. So that was, this is a fairly more recent view of this, and my children jump off this cliff all the time. Not all the time, they have many times jumped off that cliff. And I remember my 8-year-old, then 8-year-old. He was treading water next to me, and somebody, a high school student, was afraid to jump, and he called, he's treading water, and he shouts up saying, "Jump! I'm 8!" So, anyway, that is the reservoir today. Anyway, so George Soper said that's the problem, and he went upstream in the watershed feeding this river, and he found huge numbers of examples of that, basically. Even then, there were rules against doing this, and yet those rules were completely ignored. And so he found a huge number of examples of public health violations in the watershed upstream of that river. And I can't leave it there, so I'm going to go back to that one and stop. So I'll take questions. Fun, I think. Well, not for the victims, but for the rest of us. So if you went to Cornell at that time, you would say, people would say, Cornell, it's a good school, but too much typhoid. Was there a question? Yeah.
When you respond about HIV, you said those people who get infection, there's an immune response, and then it goes, is it possible that the immune response could completely wipe out the virus?